Thank you for joining us today for the 2022 BLOODPAC Summit. Welcome all of our members and all of the newcomers to our inaugural summit. My name is Lauren Lyman and I'm the Executive Director of BLOODPAC. 5 years ago, we launched BLOODPAC as a commitment to the White House Cancer Moonshot to accelerate the development, validation, and clinical use of liquid biopsy assays to better inform medical decisions and ultimately improve care and outcomes. The goal was to achieve a decade of progress in half the time, and we did it. The agenda for today and the experts we have around the table are a testament to BLOODPAC's work over the last 5 years. Our consortium members collaborate to address industry challenges within 3 pillars. Each pillar is supported by multiple working groups that define needs and ways to address them. First, we work to establish those yardsticks and guardrails that build trust and confidence in these technologies through standards and frameworks for the broader liquid biopsy community to use. Second, we aggregate data in the BLOODPAC Data Commons. This informs our frameworks and scientifically validates the work. And finally, groups work to ensure that these efforts don't sit within a silo, but rather come out of real engagement with regulatory agencies, private payers, patient advocates, and standard-setting organizations to accelerate approval and clinical use. 5 years ago, we launched BLOODPAC with 20 committed visionary members who understood that collaborating to compete offered the best path forward for the field. Today, we have over 60 members and collaborators representing stakeholders across the liquid biopsy field. The depth and breadth of our members and external collaborates adds up to create impact. Let me illustrate. Through the data contributions of organizations involved in our work, the BLOODPAC Data Commons contains over 70 studies and projects spanning nearly 4,500 cases. In all, there are almost 33,000 files and a total of 33 terabytes of data in the Commons. Throughout the day, you'll hear about our impacts, but I'd like to take a moment to highlight our peer-reviewed publications and resources for the broader liquid biopsy community. All of these can be found on our website, and I urge you to follow us on Twitter and LinkedIn as we deliver 2 new publications to the community in the coming months. This is a crowded slide, but I'm happy to say that it represents the incredible interest and expertise that has become part of the BLOODPAC effort over the last 5 years. I encourage you again to visit our website for a closer look at each of these organizations that contribute to our work. But the companies behind the work are only a small part of the BLOODPAC story. The people who have invested and shared their time, expertise, and vision with this organization are the real heartbeat of what we do. A tremendous thank you to our leadership and executive committee, our scientific co-chairs, dedicated participants from the FDA and the NCI, and finally, I want to thank every single strategic planning group participant who helped to establish the vision and projects for the next 5 years. Now that we've talked about the BLOODPAC's work and successes over the past 5 years, I would like to give a preview of the sessions over the next few hours that we'll see together. Throughout the day, we will discuss the challenges we've already tackled, offer more in-depth looks at our work and resources to date, and we will identify the next big challenges and how we plan to address them. The 3 sessions today set the stage for the challenges that must be solved in the coming years and how we'll achieve the audacious goals we have set for the global liquid biopsy community to benefit patients. Our session on MRD will address the current challenges and gaps to the implementation of MRD in solid tumors. By addressing these challenges, we can bring more tools to the toolbox for recurrence monitoring. Our session on the regulatory landscape will address how we can take our learnings from our collaboration with the FDA over the last 5 years and support alignment across global agencies. And in our final session, we'll focus on addressing barriers to clinical adoption and access for patients because we know that development and approval aren't meaningful if existing health disparities limit the full potential of liquid biopsy. A few housekeeping notices I just want to give everyone. First, we would love to hear from you throughout the day. Please feel free to ask questions via the Q&A box at the bottom of your screen. We'll make sure to respond over the next week via social media and our website. We will be answering questions live during the FDA Fireside Chat. Please put those questions in the Q&A box as well. And finally, please make sure to follow us during the event on our Twitter handle at @blood_pack and use the hashtag BLOODPAC Summit. You've heard me talk about the exceptional people who helped create and advance BLOODPAC. Now I'm thrilled for you to hear directly from them in this video that speaks to our mission, the science, and ultimately why we are doing this for patients.
Cancer Moonshot is taking advantage of how fundamentally cancer research has changed. We can do in 5 years what would take 10 years in terms of treating cancers, curing some, making others chronic diseases.
We have spent a huge amount of time creating standards, collecting data to support those standards, and accelerating the time frame to get this technology into patients' hands.
No single entity can do that. And BLOODPAC has always been that safe space where different stakeholders, from patient advocacy, from the government, individuals within the FDA, from commercial entities, academic entities, say, what can we do together that we can't do separately?
We brought together these players for the first time. The common vision is our mission statement: to accelerate the accessibility, the commercial implementation, and application of liquid biopsy tests in record pace.
BLOODPAC has a role to play in setting the standards and setting the approval paths and making sure the patient has the best benefit of that opportunity.
What if we created an ecosystem together so that we could all analyze the data together? Learning from that is how we began building what we called a data commons.
The data commons is a way to more efficiently gather, manage, and analyze data to make advances. Multiple organizations working together in a pre-competitive way need a platform to manage the data and to analyze it.
We have commitment from every member of BLOODPAC to contribute to those data commons. And now we have a growing number of committees that are looking at what are the best questions that we can ask and answer through access to that data.
The NCI participates with BLOODPAC. We all have to agree on what are the standards by which these tests are done. Would a test done in Houston give you the same result as a test done in Manhattan, right? This is really essential.
The minimum technical data elements, that is looking at the pre-analytic steps. We need quality, we need to have assurances, and the ability then to have standardized data in one place in the data commons available to everybody.
These controls or contrived materials really come into play in these types of verification studies. To have confidence that these assays are working the way we need them to, but also treat these clinical liquid biopsy samples as the precious samples that they are.
The biobank now tries to design the right protocols, standard operating procedures, how you manage, how you collect, how you process, your sample to make it fit for purpose. We've taken ourselves to the highest level of perfection, for this industry.
Physicians and payers want a lot of data. They want good data, analytic validity, and clinical utility. You've got to prove that your test works.
The growing number of regulatory approvals in the liquid biopsy area and the versatility of the technology is really driving the more and more clinical trials. The OCE learns about the state of the science in liquid biopsy technology and can also offer regulatory perspectives that can advance the field.
One of the areas of focus for the next 5 years is molecular residual disease.
Early detection of cancer. How can you say that it's completely eliminated? That can be important knowledge, one, from the drug development point of view, but from a patient point of view, when do you stop treatment? Each of those are important contexts that are best addressed in blood.
Cancer, it's a global disease of immense proportion. For all of us, it's to make precision medicines more available and do it faster than we would have done sitting in our own shops and working on it alone.
We've set the stage for another 5 years of incredible work together, moving towards the clinical validation and the utility and the accessibility out of the clinic and into the homes.
In 5 years, I'm hoping that technologies like these are going to be no different than how we think about getting a rapid test or a COVID test and being able to give not just data back to a patient, not just results back to a patient, but really insights back to patients.
The beauty and success of BLOODPAC comes from the pure dedication of our participants in taking off their corporate hats to accelerate the development and approval and really and truly the accessibility of this technology for patient care. That was terrific. All of the videos you'll be seeing today were directed by Wondros, who helped us develop and narrate our story and present this event to you today. Our first presentation now is on the state of the science in navigating molecular residual disease. It will be given by Dr. Annie Martin, Senior Vice President and Global Head of the Experimental Medicine Unit in Oncology Research and Development at GlaxoSmithKline. In this role, she leads the global team's effort to expedite the development of innovation therapeutics for treating various oncologic and hematologic indications. Dr. Martin is a BLOODPAC Executive Committee and founding member. Annie, I'm going to hand it over to you now.
Thanks so much, Lauren, and thank you to BLOODPAC for inviting me to speak today. And as was mentioned at the top of the event today, there is the ability for you to ask questions in the Q&A function. Unfortunately, we can't answer any live in this session, but we will be following up after the meeting. So it's my job today to introduce to you some of the work that we are starting to launch as we head into the next 5 years of BLOODPAC. And it's really about finding even more ways that we can advance cancer care for patients during and after treatment. And so the molecular residual disease, or MRD group, is the group that's taking the charge for this. So as we were thinking about what we would do in the next 5 years through BLOODPAC, we started to think about the different challenges that we still need to tackle. And one of those is to both identify and address barriers to how we can implement MRD in solid tumors, and also to have resources and projects that can help us to address these challenges in the long term. And so one of the goals in the next 5 years for BLOODPAC is to validate MRD in solid tumors as an early endpoint. Now, the BLOODPAC is very well positioned to do this, and you will have just seen in the video and the introduction from Lauren that we've actually accomplished a lot in setting ourselves up to be even more successful in the next 5 years. This is just a summary of some of the accomplishments that BLOODPAC has delivered to the community. And whilst we have a table here that goes from one item to another, I'm going to start from the bottom up, because really it was the establishment of the data commons in the first instance, a house that we could bring data in a standard format and make that publicly available to the community. One of the next projects that we tackled was to identify a common set of minimum technical data elements in the pre-analytic space, and to use those as a means to advance the community so that we could have a standard set of data elements that could be incorporated not just into the data commons, but across all of studies that we bring forward as the industry and the community advances. The JFDI working group also has successfully delivered and developed and characterized reference materials for cfDNA to help in genotyping of NGS assays. And finally, and most recently, the MRD Analytical Validation Working Group set forward a generic analytical validation protocol for MRD assays and have done this in collaboration with the FDA and sought feedback throughout the entire process. So you can see that we've already established a great foundation, and now we want to leverage that foundation to build upon the next 5 years. So this is essentially the roadmap that has been developed through the MRD Strategy Working Group. And one of the first challenges we feel we need to tackle is recognizing that we don't have a common language amongst the stakeholders working in the liquid biopsy space. And so the goal is to develop a lexicon with all of the key stakeholders, such as the FDA, the NIH, Friends of Cancer, and other organizations, to help us come with that common language that can then be published and shared amongst the liquid biopsy community. Just as the JFDI working group has established standards in one area in this space, we're also looking to them to help us to establish standards that are MRD-like, that can be used with different assays and really provide that quality that we're looking for across the different studies that are brought forward in the coming years. And as we move forward in the next couple of years, we also recognize that there are key data elements that we need to agree on that are important in when we report data, whether that be in publications, whether we collect the right data in our clinical studies, as well as submit those data to the regulatory authorities. And so BLOODPAC will take the lead on doing a retrospective evaluation of the literature and publicly available data to bring forward some recommendations for what those key data elements need to be. Building on all of these three areas, we also look to the long term. And really that's about building the framework to define new clinical questions, identify novel endpoints, continue to develop those protocols for everybody's use in the beyond the industry and in the community. And ultimately our goal in the future will be to run a prospective study. In all of these instances, we will publish where possible and make those data available to the community. Now, BLOODPAC is only one of the organizations that is already doing a lot of work in this space, and we would be remiss if we did not acknowledge the partnership that we have with many other key players who are helping to define this space and advance liquid biopsies for clinical use. Those include Friends of Cancer Research, the NCI, our work with the FDA, as well as standards organizations such as AMP and CAP. And we'll continue to have outreach to additional organizations who want to work with us and help us tackle these areas. This last slide is just to show to you that the MRD Strategy Working Group is truly a great group of multiple players from pharma, diagnostics, from different organizations who have come together to try to come forward and tackle this space. And we look forward to anybody else wanting to join this working group. I would be remiss if I did not acknowledge the leaders of the working group, Andy Had and Angela Silvestro. So thank you to both of you and for the great work that's been accomplished thus far. And now it is my great pleasure to introduce to you one of our keynote speakers for today, and that is Dr. Erica Carpenter. Dr. Carpenter is the director of the Liquid Biopsy Laboratory and a research assistant professor in the Department of Medicine at the University of Pennsylvania. Following both her graduate studies as well as her postdoctoral training at the UPenn, she has assumed this responsibility, which is very much aligned with the work that BLOODPAC is doing. Prior to joining the University of Pennsylvania, Dr. Carpenter obtained her MBA at Tuck School at Dartmouth College, And she worked for a number of years as a business executive in Fortune 50 manufacturing industry. Today, Dr. Carpenter is NCI funded, and her research focuses on the identification, capture, and analysis of circulating tumor cells, exosomes, and cell-free circulating DNA from cancer patients. Her goal is to evaluate different technologies in the liquid biopsy space and to find the way to advance early detection of disease, be able to monitor disease such as with MRD, as well as identify ways to better identify patients for therapy, with an overarching goal to deliver precision medicine to patients. Welcome, Dr. Carpenter, and thank you very much for joining us today.
Thank you so much, Dr. Martin. And thank you so much for that kind introduction. So we're going to take a few minutes at the beginning of this meeting to talk about the state of the science of precision medicine and liquid biopsy. And I have a number of disclosures listed here. And yes, I will be talking about a few of these companies' work in the next few minutes. So I'd really like to frame this conversation about liquid biopsy within the context of the patient's diagnostic journey, which is represented by the cartoon at the bottom of the slide. And whether we're talking about circulating tumor DNA, CTCs, or a host of other possible liquid biopsy analytes, it's important to recognize the varying role of liquid biopsy at these different milestones. So, for example, if one considers the initial workup of a patient to actually determine whether the patient has a diagnosis of cancer, that is actually a setting at which, at least currently, liquid biopsy is rarely used. After all, and again, as represented by this cartoon, sadly, most patients present with advanced disease, said another way, at a point where their tumor burden far exceeds the threshold of standard of care imaging, and therefore liquid biopsy is unnecessary. However, there's considerable effort currently to use enhanced sensitivity of liquid biopsy testing to start to move that possibility for diagnosis to the left, and in fact, reach a point where we're able to use liquid biopsy to diagnose or assist with the diagnosis of disease that is below the level of detection of standard of care imaging. And there are, of course, a number of technologies being developed to do this, some of them commercially available. And I'm just showing you one representative example here. So here, this is a pan-cancer test based on the whole genome sequencing and bisulfite conversion of cell-free DNA. And that's to enable detection of methylation markers or epigenetic signature that allows for detection and prediction of tissue of origin for a tumor. And you can see that it's available for a wide number of cancers, with each of the blue bars in the upper left-hand corner being a different tissue type, and certainly a wide range of sensitivity, ranging from 0% for thyroid, about 11 for prostate, up to 94 for carcinoma of unknown primary. But the devil's in the details here. And certainly in this published data set, if you consider the fact that each of these bars is an average across the 4 stages of disease, I pulled out the data for you just for breast and lung as examples to show that the pair of dots at the leftmost part of those figures represents the sensitivity of stage 1 tumor detection, which is unfortunately quite low at this point. However, there is quite a bit of work at this point to try to improve these tests. And I'll just give you a couple of examples. In some really nice work coming out of the Henry Ford Institute at Michigan, they've adapted this cell-free DNA methylation approach in a tumor-specific manner in the setting of glioma detection. And using this ROC curve as an example, they've demonstrated 100% sensitivity, albeit for a small number of patients in this pilot. We, and certainly many others, have started to move toward a multi-analyte liquid biopsy development paradigm. We demonstrated recently that when we algorithmically combine the results of RNA extraction from extracellular vesicles, clinical CA19-9, KRAS point mutation detection in ctDNA, and then concentration of total circulating cell-free DNA, that combined signature was more sensitive than any one analyte alone. And importantly, this is in the detection of pancreatic cancer, including early stage disease. Truly innovative platforms, preclinical platforms such as that developed by Dr. Ken Zaret, where he used genetic reprogramming to achieve an IPS-like state, mimicking early stage pancreatic cancer and facilitating the interrogation of the secretome for identification and discovery of novel biomarkers. And then finally, in very recent work, the development using nanomaterials of an electronic nose to actually mimic the olfactory sensitivity of a dog's nose and ability to detect differential volatile organic compound signatures, depending on whether the patient, at least in the case of this pilot study, had ovarian cancer shown in the red curve, which separated nicely from benign controls in yellow and then healthy individuals in green. And yet the bulk of clinical liquid biopsy and mainly ctDNA testing is currently for therapy selection. So immediately after a diagnosis has been confirmed. And that's been driven certainly in the setting of non-small cell lung cancer, but for many other solid tumors, been driven by the increasing number of approvals. And so again, this cartoon just represents the approval of all types of therapy, including TKIs and targeted drugs for lung cancer, and how that's commensurate with the approvals shown at the bottom of the slide of 2 different liquid biopsy assays. And although these have been commercially available for a few years, one of the remaining questions in the field until quite recently was whether the addition of liquid biopsy testing for mutation detection actually improved our ability to find and treat. And so to address this, we designed a prospective study of circulating tumor DNA next-gen sequencing in a cohort of prospectively enrolled metastatic non-small cell lung cancer patients. Among the 323 enrolled, about half were at diagnosis and the other half at the time of suspected progression. And importantly, in the bar chart on the left-hand side, we measured the ability to detect targetable mutations that we have drugs for, and found that the rate was about 20%, or 1 in 5, for patients who only had tissue sequencing result, but that rose to 36% when plasma results were layered on top of tissue. And this is important because at least in this study and in others that we and others have published, tissue NGS was infeasible in almost half the patients. But you might argue that what we're looking at here is driven by noise and or false positives. And so to get at that question, we performed RECIST analysis for a subset of the patients on the study. And that's represented by the waterfall plot that you see on the right-hand side. And what we calculated, and this is important, for the patients who received a TKI on the basis of plasma alone, in other words, there was no tissue result, an 86% response rate, which is commensurate with what one would expect if tissue testing had been utilized instead. Importantly, these findings were independently corroborated about a year later in the NILE study. And so it won't surprise you that clinical ctDNA testing at our institution, as shown by these bars and in many others, has just continued to increase dramatically, driven by lung, but expanding to other tumor sites. And probably as a result of the fact that we have actually embedded in our own clinical treatment pathways the use of clinical ctDNA testing, we recently measured the number of patients in our institution, metastatic non-small cell lung cancer patients who were receiving NCCN recommended molecular testing, and found it to be a very high number of 86%. And yes, that's just a coincidence that it's the same percent as the prior slide. But importantly, that compares to only 15% nationwide, suggesting that we have a tremendous opportunity using existing approved technology to start closing the gap for personalization of medicine. And so far I've talked about prescription of TKIs, but after all, and again, if I just use lung cancer as a representative example, only about 30 or 40% of patients will have such a therapeutically targetable mutation. So what about checkpoint inhibitors and use of liquid biopsy for therapy selection. So there are some hints that that will also be possible. I'm showing you two somewhat similar studies. On the left-hand side, the MYSTIC trial, in which patients received either Durva plus Trem, or a control arm of chemotherapy. And then on the right-hand side, a biomarker study in which patients, again, lung cancer patients, received standard of care pembrolizumab-based therapies. In both settings, the 500 gene panel with about 2.1 megabases of coverage was used on plasma to calculate a TMB, or tumor mutation burden, which has been previously shown in tissue to correlate with response to checkpoint inhibitor. And as you can see here, the results are similar to that in the blood. On the left-hand side, in the left-hand Kaplan-Meier curves, you can see that when they dichotomize based on high and low mutation burden, that the patients with high mutation burden did better, and a similar result on the right-hand side. And so we wondered, is it possible to also slide that red arrow to the left, just as we're thinking of doing for diagnosis? Is it possible to detect therapeutically targetable mutations early in the diagnostic workup such that we can reduce the time to treat? To address this, Dr. Jeffrey Thompson conducted the following pilot study. He enrolled 2 cohorts of patients. In cohort 1, the patients had their blood drawn literally at the time that they were going in for diagnostic bronchoscopy. So at the time that lung cancer was suspected, but had not yet been pathologically confirmed. He compared that to a separate cohort of patients, cohort 2, that received their liquid biopsy testing according to standard of care at our institution, which is typically at the time of the first visit to the medical oncologist or soon afterwards. Not surprisingly, he noticed that on the left-hand side, 85% of the patients in cohort 1 had their genotyping results at the first oncology visit. That facilitated for 74% of those patients, in the middle figure, a therapeutic decision being made in that first visit, very reassuring for the patients. And then on the right-hand side, you can see that this did significantly reduce time to treat for these patients. And I'm going to spend the remainder of my time talking about MRD, or minimal residual disease detection, a concept that certainly has its roots in hematologic malignancies. And, if you think about the premise here, if the half-life of circulating materials, whether it's ctDNA or CTCs or what have you, is really on the order of a few hours, then, and I quote, the detection of ctDNA or CTCs months or years after curative intent treatment indicates the persistence of MRD. And to put a slightly finer point on it, imagine 2 different patient scenarios. In this figure here, a patient represented by the red curve would have somewhat decreased tumor burden in response to curative intent treatment, but that would then rebound in the form of clinical relapse, suggesting perhaps that if that process could be quantified, that there might be an opportunity to sustain or even escalate therapy. Contrast that, if you will, with the patient represented by the green curve, whose tumor burden becomes undetectable and remains undetectable over the course of therapy. Begging the question, I believe Dr. Scher posed this in the beginning video, when do you stop treatment? And when might you, with some degree of assurance, choose to start to de-escalate therapy? And so most MRD detection through liquid biopsy is currently done using ctDNA. Barriers for this have been discussed in the prior talks: low sensitivity, high false negative rate. I believe Dr. Martin talked about standardization and the need for standardization, not only pre-analytical, but for the analytics and then also the interpretation. And we'll talk a little bit more about that in a second. The lack of randomized trial demonstrating a clinical benefit, and then background noise as introduced by, for example, clonal hematopoiesis. Benefits are, in fact, that it's non-invasive testing. And that, in fact, means that it may actually be more comprehensive than tissue, and it allows for longitudinal monitoring. And please don't forget what I shared with you a few slides ago, that in some cases, For example, in the setting of lung cancer, up to about half of patients are unable to complete tissue next-gen sequencing. So starting with one of the earlier examples published in 2017 in Nature, this is a tissue-informed ctDNA-based MRD test. So resection and multi-region sampling of, in this case, a lung tumor is performed, followed by exome sequencing, and that would then inform the design of a phylogenetic tree and a PCR-based assay of several different variants, form a bespoke assay that would then be applied to the sample, the blood sample. This then allows for monitoring, and in this representative example of one of the 14 patients in this study who relapsed, you can see the different elements of MRD detection. In the red curve, you see the subclonal mutations, or the average of the subclonal mutations, which does in fact become undetectable immediately postoperatively and then rebounds. However, the clonal mutations, as represented by the blue line, decrease but remain detectable and predict relapse a couple of hundred days before that relapse is actually confirmed by standard of care imaging. And so that was one example of, again, a tissue-informed assay. This certainly allows comprehensive genomic viewing, and it yields multiple targets, as I just described. It can reduce the background from non-tumor mutations, increase sequencing depth, and also improve the ability to detect a variant allele fraction of less than 0.1%. A second category, however, increasingly being studied is that of tissue agnostic tests. And so this obviously avoids the requirement for sufficient quantity quality tissue DNA, which I've told you can be an issue, decreases turnaround time and cost, and again, may more comprehensively reflect the changing profile, especially over time under pressure of therapy. So there are very few ctDNA MRD platforms currently used in clinical practice. And so I borrowed a table here from a recently published review that actually just describes two of them. So Signatera, which you saw a couple slides ago, and then the Guardant Reveal test. And all I wanted to do with the slide was to make the point of the various comparisons. So as you can see, the Signatera assay calls variants, whereas Guardant, which I believe used to be called LUDER1 until recently, calls variants as well as methylation markers. One is tumor informed, the other is tumor naive, and yet they both achieve about the same VAF for their LOD. Notice the considerably shorter turnaround time for the Guardant Reveal assay. On the other hand, it is focused on a single tumor type, CRC, colorectal cancer, as opposed to the multi-cancer applicability of the Signatera test. And I wanted to use this figure that I pulled out of Parikh et al., in which they looked at the Guardant Reveal test. But I'm using this really to make a point that's representative of an issue of many of the existing tests, and that is the need to address the false negative rate. So if you look at this bar chart and focus your attention on the left-hand bar, you'll see that 49 patients were MRD negative. In other words, no ctDNA detected, and yet 12 of the 49, so almost a fourth of them, did in fact recur. And so this has really driven a push for improvements in sensitivity. And again, there are a number of different groups working on this. I'm showing you one example of a commercial assay on the left that will be presented at AACR, and then obviously very nice work coming out of the lab of Max Steen at Stanford on the right-hand side. Both of these tests are tumor informed and are based on whole genome sequencing of the tissue. And although they use different technological approaches, they are both seeking to achieve a sensitivity of about 1 to 3 parts per million. As far as RUO or research platforms available, the list is actually quite long. I didn't have room to put them all on here. And in fact, again, at your leisure, you can go and read the tables from this very nice review. But the point that I wanted to make here was really about the heterogeneity that continues to exist. And this is a theme that both Lauren and Dr. Martin brought up. So as you look at this table, you see different types of variants queried, different technologies used, tumor naive, tumor informed, and a very wide range of LODs. And so this sort of brings up the question of, how do we get our hands around this, and what should we do as a field going forward? So we would argue that evidence generation is an essential pillar moving forward. And certainly Friends of Cancer Research and using their CT Monitor project step 1 has really taken leadership in this area. And thank you to Mark Stewart for providing these images from a manuscript that ought to be in press within a few days. But just to preview you on that, they have worked to generate evidence that changes in ctDNA levels may accurately reflect therapeutic effects. To do this, they gathered data on lung cancer patients with metastatic disease who had received checkpoint inhibitor therapy and had 2 or more liquid biopsies available. This was done across 5 different institutions, of which Penn Medicine was one of them. And importantly, this adapted swimmer plot really says it all. If you consider, so the blue stars are the timing of RECIST, but all of the other colored circles are representative when liquid biopsy was ordered. And so you can see the sheer level of heterogeneity in terms of not just the number of times it was ordered, but also the frequency and timing. And so they sought to answer the question of whether the trends that are observed in a smaller independent data set can be replicated in a larger combined data set. And as you can see in the Kaplan-Meier curves on the right-hand side, whether one dichotomizes based on decrease or increase of ctDNA or a 3-step analysis, there is a very strong association that when ctDNA comes down, that tends to predict better overall survival. Under the heading of continuing to generate evidence, we need to keep cycling back to what the prior standard of care was. In this very nice study that was just published a couple days ago, they enrolled 48 patients who had their colorectal cancer surgically resected, and then compared ctDNA testing for MRD detection to imaging alone or imaging plus CEA. And as you can easily see by the overlapping black and yellow curves, there was actually no difference here, suggesting, as the authors said, that the ctDNA test, at least in this case, may not provide advantages as a surveillance strategy compared to standard imaging and CEA. An additional pillar would surely include the application of testing in rare and low-shedding tumors. A lot of the work so far has really been concentrated in lung, breast, and colorectal cancer, but we and others have certainly started to push out on CNS tumors. I'm showing you some very recent work, and if any of you were at the session yesterday, you would recognize that this figure was presented by my colleague, Steve Bagley, and we're using it to make the point that even in the setting of a very low shedding tumor, when using the technology that was recently published in Nature Medicine by our partners, that we can actually calculate a tumor fraction. And when we dichotomize based on high and low fraction, you can see the very strong association here with survival, even after correcting for clinical factors. And then finally, a pillar that echoes what Dr. Martin said and includes the rigorous assessment of the liquid biopsies in the setting of a clinical trial. As one brief example, in this randomized study of adjuvant atezo, and this is in the setting of operable urothelial cancer, these authors sought to determine whether MRD positive patients who have a high likelihood of recurrence would derive a clinical benefit from adjuvant checkpoint inhibitor. And as you can see in the unselected patient population, there's indeed no difference between the atezo and observation arm. However, within the cohort of patients who were positive for ctDNA, you can see a large and significant separation of the 2 treatment arms. And that suggests, of course, that once the prognostic association has been established in trials such as that in the green arm, that we might move to intensification trials, design strategies, as well as de-escalation. And last, but certainly not least, I wanted to mention the use of other technologies such as circulating tumor cells and starting to move beyond ctDNA. In this success phase 3 trial of adjuvant chemo in the setting of high-risk breast cancer, early stage breast cancer, these investigators use cell search to enumerate CTCs at year 2 after chemo. And as you can see by the separation of curves on the left for OS and DFS, this was indeed associated with a difference in survival. And as you can see on the forest plot on the right, that held up for most subtypes. So this is a really brief amount of time to cover an enormous amount of real estate in terms of the current science of liquid biopsy. But by framing it within the patient's journey, hopefully I've convinced you of its relevance. And although some of the takeaways include early detection, currently lacking sufficient sensitivity for early stage cancer detection, there is an enormous amount of work done, and we remain very optimistic that these tests will achieve that. Certainly, liquid biopsy is an essential component of tumor profiling for personalized therapy. And then we have this emerging world of MRD, which I'm very excited to be a part of. Several technologies that are under development that I've described, but it will be essential that these are validated in interventional trials going forward. I thank you so much for your attention. I'm really looking forward to participating in this meeting. Thank you.
Thank you so much, Erica and Annie. We really, really appreciate your time and expertise. The next video we are going to show highlights our work developing frameworks and standards, an essential building block for fostering trust and clinical adoption of these novel technologies. We have already created frameworks for analytical validation, pre-analytical minimum technical data elements, and evaluation of reference material, but there is still much to be done. Here are our BLOODPAC members discussing the importance of standardization and our ambitious vision for the future.
If we have a standardized approach to doing analytical and ultimately clinical validation, then we can compare apples to apples data.
One of the things that is hardest to do is to develop standard operating procedures for various testing methodologies, so you get the same answer no matter where the test is done. And I think BLOODPAC has contributed significantly to that effort.
Now that's really important because we need quality. We need to have assurances that if you're going to compare data across platforms, that you have the confidence in those data because you've established a common set of standards and everybody's working with those standards.
These technologies are complex enough where a lot of collaborations of multiple players is helpful in establishing what are the acceptable standards for validating such technology, for demonstrating its accuracy and precision and other characteristics.
Yes, we have commercial entities that are going to be bringing incredible technologies to market, but we also have the academic thought leaders who are participating. We have the government agencies that are participating so that we're working together across those perspectives.
This collaboration platform really allows the BLOODPAC to create high quality guidance about factors that can influence the performance of liquid biopsy assays that are really likely to be adopted by the community.
And that's why a group like the BLOODPAC is important when you have all the players coming together to speak in one voice.
An exciting potential future use is the ability to potentially detect molecular residual disease after treatment, which could really help inform whether a patient should receive an additional or a novel therapy.
And as we focus on the next 5 years, launching a control material study around for MRD is going to be very exciting and very complex, but I think worthwhile.
Standards help you evaluate the information in front of you and know that, well, if it followed these guidelines, we have more confidence in the one little nugget of information that's useful and valuable and valid.
The work that we're doing at BLOODPAC, setting these standards, will ultimately provide credibility to everyone, to the field in general.
Thank you to all of our members who helped capture that story. Our next presentation on global regulatory challenges and opportunities will be led by Dr. Hakan Sakul, Vice President and Head of Diagnostics for Pfizer and BLOODPAC Scientific Co-Chair. Dr. Sakul led the implementation of Pfizer's diagnostic strategy and currently directs its diagnostics program. Hakan, we are so looking forward to this discussion.
Thank you very much, Lauren. So today I will speak to global regulatory challenges and opportunities on behalf of the Global Regulatory Strategic Planning Group. BLOODPAC has done a wonderful amount of work and actually quite ambitious amount of work completed already in the first 5 years. And as we look into the next 5, I think it's very important to think about regulatory challenges and how to work together. Since the inception of BLOODPAC, it has worked with the FDA. And but today the challenge is that beyond that work, there are only a few regulatory approvals and no international regulatory standards. And you've already heard multiple times developing standards, developing minimum technical data elements, and you'll hear about clinical utility as well. But at the end of the day, all of these are going to be benefiting the patients. So the patients are really front and center. And for that to happen globally, we also need to make sure that these standards are also socialized with global regulatory agencies. And that really is the challenge that this particular working group is working on. And so our 5-year goal is to facilitate the harmonization of guidelines and expectations for review of liquid biopsy technologies so that we can enable rapid approval to benefit those patients globally. So here's a selection of initiatives and to show you some of the impact in the field. And for the pre-analytical variable working group, they've identified and published a common set of minimum core data elements. And you've heard about that already. And the analytical validation working group has worked on alignment of generic analytical validation protocols with FDA feedback through official pre-submissions. And the patient and clinical context variable working group is working to establish an agreed upon list of disease-specific and patient-specific variables that may impact the outcome of an assay using the FDA pre-submission process. And you've already heard about the data commons that has established an open and publicly accessible database for the global liquid biopsy community. So all these successes put together and those that are still in the work really position us now to focus also on the global regulatory collaboration to enable all these impact to really go more global and again, with the patient and being front and center, be able to make that impact on the patient. There are a few global regulatory themes that we've worked on through our intake meetings in some of the initial regulatory sessions. And we certainly have established interest from all groups that participated in these intake discussions, and they have expressed interest in collaborating with BLOODPAC. And they have certainly have interest as well in minimal technical data elements for MRD trials to define the clinical purpose, and also to work on data needs to define MRD as a relevant secondary endpoint. But there are challenges. One of those is sensitivity. That is looking into absence of ctDNA versus sensitivity of assay. There are challenges in clinical and non-clinical sample limitations, but those challenges also create opportunities for us to be able to pursue. Reviews to date use tissue criteria for liquid biopsy submissions, and there are lack of reference standards, and that is a barrier that we feel we can work on in the US and globally. So with that in mind, our short-term goal is to establish a global regulatory partnership, which is a lot easier said than done. We do appreciate particularly the help of the FDA for us to be able to reach out to other regulatory agencies globally. And you'll see a list shortly. And another short-term goal is then to obtain regulatory input from these regulatory agencies globally on our current BLOODPAC deliverables. And in the midterm, seeking additional deliverables so that we can solve these global challenges identified by those partners that we hope to join us, and then develop those deliverables recommended by those partners. And in the long term, to implement those deliverables to support regulatory submissions, again, globally, so that we have common standards and common criteria to consider. So we've done intake discussions with a list of agencies and organizations shown here. And we have more coming, and I'd like to specifically recognize those here. The notified body, BSI, the UCOPE, trade organization in Europe, and then regulatory agencies in the US, Canada, Israel, Japan, and in Switzerland. And we have more of these coming. We hope we will be able to create a global regulatory partnership that'll work in tandem to be able to implement some of these solutions that BLOODPAC has come up with in terms of data standards and in terms of critical elements to consider for the approvals. I'd like to also recognize the participants in the Global Regulatory Strategic Planning Group. And they're shown here: FDA, PGDX, Natera, and Tempus have been significantly contributing to this program. And I think that is my last slide. And with that, I would like to introduce, so the next presentation is actually a fireside chat. And that's a conversation that Elaine Katrivanos is going to moderate. So let me introduce her briefly here. Elaine is Vice President for Regulatory Affairs at Tempus Labs. And in this role, she's responsible for leading the Regulatory Affairs Department. And prior to joining Tempus, she was a regulatory lead in pharmaceutical drug development at AstraZeneca. And before that, Elaine worked at CDRH at the FDA for 12 years and served in a variety of key roles, including pre-market and post-market device review, policy development, and management. Elaine holds a degree in biomedical engineering from the University of Pittsburgh. And now I'll pass it on to Elaine to introduce her guests, as well as introduce the fireside chat. Thank you.
Thank you, Hakan. It's my pleasure to introduce two leaders in liquid biopsy and therapeutics at FDA, Dr. Julia Beaver and Dr. Rena Phillip, who will be participating in our fireside chat today. Dr. Julia A. Beaver is the Chief of Medical Oncology in the Oncology Center of Excellence at the U.S. Food and Drug Administration, and the Deputy Director acting in the Office of Oncologic Diseases in the Center for Drug Evaluation and Research at FDA. She was previously the Director of the Division of Oncology 1, and prior to serving as Division Director, she was Breast Cancer Team Leader and then Supervisory Associate Division Director. Through her time at FDA, she has spearheaded the authorship of multiple FDA guidances for industry, including guidances related to the expansion of eligibility criteria, male breast cancer, placebos and blinding, and metastasis-free survival. Dr. Beaver has also published multiple regulatory research projects and co-leads in the OOD-OBP Next Generation Sequencing Translational Research Laboratory. She serves on the Liquid Biopsy Blood Profiling Atlas in Cancer, or BLOODPAC, as we know, committee, and has particular expertise in precision oncology. Dr. Beaver is also an assistant professor of oncology part-time at Johns Hopkins University, where she's a member of the Johns Hopkins Breast Cancer Group. Dr. Beaver graduated magna cum laude from Princeton University and then earned her medical degree from the University of Pennsylvania School of Medicine. She completed a residency in internal medicine at Johns Hopkins University School of Medicine, followed by a fellowship in medical oncology at the Johns Hopkins Sidney Kimmel Cancer Center. Then turning to Dr. Phillip. Dr. Phillip currently holds the position of the director in the Division of Molecular Genetics and Pathology in the Office of In Vitro Diagnostics, also known as OHT7 at the Centers for Devices and Radiological Health at the FDA. The Division of Molecular Genetics and Pathology provides regulatory oversight of in vitro diagnostic tests in a variety of product areas, including oncology molecular tests, pathology and cytology, companion diagnostic tests, and genetic disorder molecular tests. At the FDA, she has been involved in many diverse activities, including pre-market clearance and approval, manufacturer assistance, post-market regulatory compliance actions, and helping with drafting several guidances. In addition, she has been an ongoing participant in several biomarker harmonization efforts with external stakeholders and actively working to develop regulatory frameworks for any new challenging in vitro diagnostic assays in oncology. Prior to joining FDA, Dr. Phillip's experience included 3 years at the US biotechnology industry. Dr. Phillip received her PhD from the University of Illinois at Urbana-Champaign. Now I'd like to get started with the discussion. Please feel free to submit questions using the Q&A feature in Zoom, and we will endeavor to answer these questions at the end of the session, time permitting. Rena and Julia, both of you are in a unique position to have seen many regulatory applications related to liquid biopsy and therapeutics. BLOODPAC aims to support these regulatory applications in multiple ways, including several pre-submissions to align with the agency on baseline elements for these types of files. In an effort to ultimately get useful tests and beneficial therapies to patients as quickly as possible. From your perspective, what impact has BLOODPAC's work had on the applications you've seen? And I'll start with Rena, kicking this off from a CDRH perspective.
Thank you, Elaine, Lauren, and the summit organizers for inviting me to participate in the summit. So we at CDRH have worked with BLOODPAC, I would say, from its inception in 2016. We support the goals of the BLOODPAC, especially bringing the liquid biopsy community together, developing expectations for minimum data elements that are needed for liquid biopsy sample collection, that is the pre-analytical variable part, then moving on to the analytical validation and continuing into the patient context variables that will drive the clinical validation of liquid biopsy tests. So through several pre-submissions, as I think the previous speakers have already alluded to, BLOODPAC has been working with FDA, and we have worked with them on these deliverables, that is the minimum technical data elements for pre-analytical variables, analytical validation protocols for targeted NGS oncopanels, and the manuscripts from these two projects, where there's also very good collaboration with many stakeholders, including the FDA. So we believe that these two documents, especially the analytical validation manuscript, has had a good impact in the liquid biopsy field. In fact, we make test developers aware of BLOODPAC's publications during our pre-submission process, and we are looking forward to further deliverables from BLOODPAC. We are hoping that they will shape the liquid biopsy field.
Thanks, Rena. Julia, do you have anything to add from a CDR perspective?
Yes, sure. And first, a really big thank you to Elaine, Lauren, the organizers for inviting me to participate. I've been fortunate to be a part of BLOODPAC with Rena since the initiation of the effort in 2016 as part of the initial moonshot, and have really seen the impact of BLOODPAC grow over these last years. And Rena mentioned many of the important contributions that BLOODPAC has made. And I'll add from a clinical standpoint, as I supervise drug application reviews, understanding analytical validity and standardization of assay characteristics really has allowed for more confidence in trial initiation and ultimately more confidence in the clinical data that gets generated. And particularly over the last year or two, we've seen more applications incorporating ctDNA, more clinical trials, and more clinical applications relying on ctDNA as assays to select patients. And I have no doubt we're going to see only more of this as this area is very relevant going forward with many other potential uses I'm sure we'll get into later in this chat.
Yes, certainly. We definitely are interested in talking more about that and maybe pivoting from regulatory applications into clinical use. What benefits and challenges would you expect in implementing liquid biopsy testing into clinical practice guidelines globally? And Julia, I think I'll start with you, and then Rena, if you have anything to add, feel free.
And I think some of the speakers earlier mentioned this. The most common current clinical use for liquid biopsy includes ctDNA testing for selection of patients for biomarker subgroups that would be expected to respond to a given drug. And for this, we have multiple approved companion diagnostic tests that are part of clinical practice guidelines for this use, really ranging from non-small cell lung cancer to ovarian cancer to breast cancer, and also tumor profiling claims. And this use can be particularly helpful clinically in situations where tissue is more challenging to sample. Some of the challenges or limitations are with the sensitivity. So if a positive ctDNA result, for instance, the treatment can be confidently selected, but if a negative result, reflex testing to tissue is recommended to make sure no positive is missed. And of course, there are many other potential uses for liquid biopsy through the whole spectrum of a patient's disease course, which may require more concrete evidence in order to incorporate into guidelines globally, starting with early diagnosis of cancer and more of a screening paradigm to adjuvant uses, as touched on, molecular residual disease, understanding resistance, and really require further studies and data to be more widely accepted for clinical practice guidelines.
Thanks, Julia. Rena, did you have anything you'd like to add? Yeah.
Maybe just a few points from device perspective. The challenges of liquid biopsy tests continues, right? The false positives, false negatives could be addressed, but the false negatives Julia already mentioned. That, for example, if the tumors do not shed, it could give false negative results, which could be addressed by the reflex testing to an FDA-approved tissue test. But there is also sometimes you need to be addressed about the false positivity test, because sometimes these tests have very high analytical sensitivity, and that could give rise to some false positive results, which needs to be addressed. Also, as the previous speakers already talked about, the chip mutations could also give rise to some of these false positivity results.
Thank you. So focusing on a specific type of liquid biopsy that we had mentioned earlier, molecular residual disease, or MRD. Rena, I'm curious about your thoughts regarding the opportunities and challenges regarding MRD liquid biopsy testing.
Again, I guess we heard the previous talk was really excellent. Talked about how the use of ctDNA as a promising marker, the use of MRD in the management of cancer at all stages, right? And so, it could guide adjuvant treatment, it could predict relapse early, it could help to monitor response. There may be lead times compared to imaging, which may give an earlier readout on patient status with a given condition, and that could allow for proper intervention. However, there's a lot of groundwork that needs to be done on the technical, on the analytical, on the device, and to make the most of these MRD clinically. In particular, there are some, as I think the previous talk already mentioned about, there are ongoing working group efforts to study changes in ctDNA and the early stage disease. In the assays that are used in these efforts, they need to be harmonized if you have to generate poolable data. And when we talk about assay, MRD assay validation, It starts from it's the entire assay system. It's from the sample collection to the output of the assay, which includes the cutoff, the cutoff that determines the positive versus negative. So, because it starts from the collection, the standardization of the pre-analytical steps, that is the blood collection, handling, processing, all those are critical when you are evaluating MRD. And then the cutoffs, the device cutoffs, those should be analytically and they should be clinically justified. There should be a harmonized, clinically valid definition of the MRD cutoff. So there needs to be a lot of work done on that aspect of how do you define the cutoff for MRD. There needs to be a lot of work on the timing of ctDNA measurements, because the timing, the shedding of the ctDNA is affected by the histology, the grade, the stage, the size of the tumor. So therefore, the timing should be supported by the performance characteristics of the test. And also there should be studies to look into the multiple time points. And all of these studies, all of these issues I addressed should be looked into by good studies that maybe the BLOODPAC could initiate some of these, which will be helpful for the liquid biopsy field.
Thank you, Rena. Julia, from your perspective and experience with clinical studies, what expectations do you have for MRD testing in clinical trials specifically?
Well, I think this is one of the most exciting potential uses of ctDNA, particularly in the adjuvant or neoadjuvant setting, to use ctDNA as a marker of molecular residual disease. And we know that, and as mentioned earlier, across many cancer types, the presence of ctDNA after adjuvant therapy or surgery can portend a poor prognosis. And we can really use this information to enrich a clinical trial for that higher risk population. And that would need to be done prospectively. And historically, adjuvant trials take such a long time, require thousands of patients to be enrolled. But by using this enrichment design as an enrollment criteria, there's the potential to select this more high-risk population that could benefit from an additional intervention that could be tested and require a smaller sample size in a shorter time frame. And not only escalation trials, but also de-escalation trials could be done in this space, which is really important for advancing the field. We overtreat patients in the adjuvant setting. It's an unfortunate fact. And so the idea that you could select a patient population by ctDNA in a de-escalation trial to really de-escalate adjuvant therapy would be a great benefit to the field and improve toxicity and long-term toxicity for these patients who ultimately could be cured. But as Rena mentioned, in order to implement this successfully, we need to make sure that the sampling considerations and analytical validation studies are appropriate. And this is a space definitely where BLOODPAC can help with providing information in support of this.
Thank you, Julia. And as we've talked about here and in other sessions, liquid biopsy diagnostics and therapeutic product development often go hand in hand. Rena, from your perspective, how has liquid biopsy diagnostic testing influenced therapeutic product development with an ultimate goal of getting these therapeutics to patients as quickly as possible?
I think Julia touched base earlier a little bit, but I can still say some of the history and how things have been in this liquid biopsy, because we continue to see many clinical trials still enroll based on tissue-based testing. But these trials are also collecting the blood samples for future testing with ctDNA-based testing. So our first liquid biopsy approval was such an example, right? The cobas EGFR mutation test that was already approved using FFPE tissue specimens as the companion diagnostic test for detection of exon 19 deletions in L858R for non-small cell lung cancer for TERCEVA. And then, but they had also collected samples, plasma specimens from the same clinical trial, and that supported the efficacy of the tissue-based test. So that was used for testing with the approved liquid biopsy test. So depending on the PPA and NPA, based on the comparison between the tissue and the liquid biopsy test, there is always additional labeling languages, such as the reflex testing to tissue if it's really high NPA and lower PPA.
Always comes down to the statistics. Julia, do you have anything to add to that?
Just that it's the growing number of the regulatory approvals in this space, and the advancing technology is really driving the use of the liquid biopsy assays in a greater number of clinical trials. And these studies can lead to additional future applications of liquid biopsy in patient and clinical settings. And as I mentioned earlier, there's really the potential across the whole spectrum of disease to incorporate liquid biopsy. And one use we haven't touched on fully yet that I know is of great interest and was mentioned earlier in the session is the potential to use ctDNA as an endpoint to predict clinical benefit. So this would be different than the prior use I discussed, different than selection of a patient population, but this would be the potential use as an approval endpoint. And this change in ctDNA would be seen more in a treatment arm versus a control arm, and that has the potential in the future to be used to support drug approval, although we don't have the evidence yet to support this use. But organizations like BLOODPAC, public-private partnerships, are really key in developing the evidence necessary here. And some of the efforts mentioned, like the lexicon, the reference material data elements, will really assist with understanding those analytical and performance characteristics that could be more reliably used for this context of use. And moving forward into thinking of clinical questions, prospective evaluation will be key. And I think collaboration with other efforts, as mentioned, such as Friends of Cancer Research CT Monitor, which is also investigating in this space, will be key as well.
Yeah, that actually leads to my next question, which was, what will it take for FDA to approve a precision therapeutic with liquid biopsy testing only? It seems like you've addressed a lot of that. Do you want to touch on anything else, Julia? And then I'll turn it over to Rena to comment.
Sure. Well, I guess taking a step back, we have approved in the prostate cancer space liquid biopsy tests only, kind of without the corresponding tissue tests. But I think you're asking more as that endpoint, use as an endpoint. And I think we have some of the examples of that sort of perhaps corollaries, thinking about pathological complete response in breast cancer and the amount of evidence that was generated to support those approvals, really thinking about larger meta-analyses coupled with prospective data studying those endpoints, and importantly, standardization of the definition of that pathological complete response. And so I think those will really be important as we think about how to use liquid biopsy testing as an endpoint to support drug approval.
Yeah, so Julia mentioned about the prostate cancer, the approval, but most of the approvals we have had, so if it's negative, it should be reflects to a tissue test if possible, right? So that's what, but I guess if I can see the community is looking into what can it take for an approval with only ctDNA only, right? I mean, we are seeing some trials because, as I think Julia mentioned earlier, if the tissue biopsy is really difficult, there may be the primary enrollment could be based on ctDNA. But from a device perspective, in order to approve with liquid biopsy testing only. The liquid biopsy test must have a sufficiently high PPA to tissue because the patients who may benefit from highly effective drug should not be missed, right? We all agree with that. And this could be the cases where the tumor biology is also well studied, and we know the shedding rates are high, and so therefore the tumor and the tissue results are comparable. And I guess maybe this is another area. Maybe I'm hoping BLOODPAC can create a database with this information for multiple tumor types with this tumor tissue and blood sample comparison that could be useful for these kind of uses.
Thank you, Rena. We'll certainly take that back. Switching gears a little bit, an important area of when you think about improving public health and patient care is addressing unmet needs. What unmet medical needs could liquid biopsy testing help address? And I think I'll start with Julia.
Well, because by definition, liquid biopsy is relying on analyzing blood samples, which are more easy to collect than tumor tissue, liquid biopsy testing may be more easily accessible to patients, particularly those treated in diverse care settings, to inform not only treatment selection, but perhaps more rapidly allow for enrollment onto clinical trials. And because it's relatively more easy to collect blood samples at multiple points during a patient's care than, of course, tissue biopsy, liquid biopsy can also provide that ongoing information about which precision therapies might be most beneficial based on the molecular characteristics and evolution of the tumor. And one of the efforts we've been really involved in the Oncology Center of Excellence is related to decentralized clinical trials, and the idea that liquid biopsies could fit more readily into this paradigm is very attractive.
Thank you. Yes, we definitely want to consider these aspects when we are, thinking about liquid biopsy and therapeutic product development. It's very important. And I think this will be one of my last questions. Given that BLOODPAC is interested in supporting FDA and bringing high quality tests for liquid biopsy and therapies to patients as quickly as possible, and I think we touched on some of this, but what should BLOODPAC keep doing? What else should we do? And what can we do differently in an effort to accomplish this goal? Maybe Rena, I'll start with you and then I'll turn it to Julia.
Sure. Yeah, as I mentioned at the beginning of our chat, we strongly support the goals of that BLOODPAC, bringing together the community and developing expectations for the pre-analytical, analytical, and clinical validation for the liquid biopsy testing. These community input and involvement, they play a key role in the development of standardized SOPs and protocols, and also in the harmonization of biomarker. And also, there are other assay definitions that could be harmonized, as I mentioned earlier, like the MRD cutoff. So this engagement of multiple stakeholders is very important, contributing to this. And these direct engagement, it is also a critical source of information about how assay validation approaches, challenges that are encountered with this validation of novel biomarkers. I mean, there are these complex biomarkers like HRD, MRD, TMB. There's a lot of work to be done in these complex biomarkers. And there is a lot of challenge still sourcing samples for validation. So, coming up with strategies to mitigate these challenges and developing performance expectations for tests that are needed for making these important healthcare decisions. So, we continue to look forward to more projects, such as the development of MRD, BTMB protocols, reference samples covering different variant types, as well as what I mentioned earlier, the plasma tissue comparison across tumors. across multiple tumors, so that those could serve as a common database that we could use if there is a trial that is only enrolled using ctDNA-based testing.
Thank you both. I appreciate that. And now I'd like to turn to my colleague, Dr. Jennifer Dickey from PGDX, to share some of the live questions that we've received. There you are, Jen. Great. Would you like to pose some questions to Julia and Rena?
Absolutely, and the audience can continue to add questions in the Q&A section, and I'll start asking them. So this one is to both of you. We have discussed several times challenges about the sensitivity of liquid biopsy assays, and we've also talked about multiple clinical uses, including treatment selection, disease monitoring, and others. Is there an FDA recommendation of how to set like minimum sensitivity requirements around these clinical uses?
Jen, is the question, is there a FDA recommendation?
Yeah, about sensitivity needs for clinical uses?
It depends on the use, right? For different uses, you will have different needs. And for MRD, the sensitivity should be really low so that you can pick up, but at the same time, it shouldn't be false positive. So that's the balance. And that's what I was talking about. What is the cutoff? How do you define the cutoff? And so, the answer is we don't have a straight answer to this, but it depends on the intended use and how it's going to be used in the real use.
And just to add, this is something that we discuss with companies throughout their drug development program as they start getting information based on the testing, and ultimately that we review during the drug review that's submitted for approval to decide, is there a reflex testing that should be added to the recommendation or not.
Great, thanks. And we have discussed during this in the last question, a need for liquid biopsy to be concordant to tissue biopsy, and when we should expect a high level of concordance. But are there instances where you might not expect that concordance? And for example, if the tissue biopsy is less representative of the tumor environment than blood, or when tissue biopsy is not a feasible approach, what would be the FDA expectations then when tissue concordance can't be obtained or isn't representative?
Well, I can start this one, and then Rena can add on. It's very similar to the last answer. It really depends on the situation. We always appreciate having that additional information, having the tissue and having the liquid to be able to look at the performance of both if possible, and to be able to make some of those assessments. Is perhaps the liquid biopsy as a selection more resulting in a better efficacy of the drug, or is it the tissue, or is it what we've typically seen is it's both, and the liquid does have some false negatives. Therefore, we have given the reflex recommendation when we have the drug approval.
Right, and also there's been another approach, right, when the NPA was pretty also low, which means you don't know whether the tumor negative, plasma positive response, how do you address that? And there have been some post-market commitments to look into that aspect, whether that is really holds true in the real world setting.
Okay, great. So we've been focused a lot on liquid biopsy for mutation detection, but some members of the BLOODPAC are focused on other markers within liquid biopsy. And so, for example, if you're looking at methylation-based assays, Will there be different standards of approval or different regulatory guidance for assays based on methylation than those that are simply looking for DNA mutation?
No. So the pre-market approval will always depend on the intended use. The risk of for example, if it's companion that's regulated as a PMA versus if it's a monitoring assay. So if it's a methylation assay, but if it's still used for a companion, yes, it could be regulated as a PMA. So it depends on the intended use, right? And what are the analytical validations? So those are I didn't bring that up in my answers. Pre-submission is the answer to if you have questions on how FDA is going to look into the analytical validation of methylation assays, please submit pre-submission to us, and we will discuss during the pre-submission process.
And I would add that the agency has been very helpful in BLOODPAC's pre-submissions. BLOODPAC has submitted several pre-subs and received a lot of useful feedback to guide the baseline characteristics for various aspects of liquid biopsy submissions.
Thank you, Elaine.
Absolutely. So now we have a question about minimally invasive blood collection techniques, and particularly in the pandemic environment, making use of blood collection techniques done by patients themselves at home. Has the, either for pre-screening or long-term follow-up by disease, has the agency been considering or been in discussions about what would be needed for at-home liquid biopsy testing by patients?
Well, I think this is more of a question related to some of the analytical aspects, but from a clinical trial aspect, that goes along with what I mentioned earlier about decentralization, easing the burden of trials or clinical care. So in theory, it sounds very good, but that's something that would need additional vetting with CDRH. So I don't know, Rena, you can comment on what that would be.
Yeah, I wasn't sure, Jen, your question was about sample collection at home, whether there have been there have been a lot of discussions on that, especially with the COVID situation. There have been several working groups working on it. I mean, like at least one major working group working on what are the requirements needed for sample collection at home, because that's all altogether a new territory in terms of the pre-analytical steps that are needed, and how do you make sure that there is no, there are lots of missing issues can be associated. So there have been some working group that is working on that, especially on that. Yeah, maybe just in one cancer to begin with, and could get to the others in the future. Yeah.
That's great. So the BLOODPAC Analytical Working Group has proposed that LOD as a measure of sensitivity is not appropriate for complex biomarkers such as BTMB, or, BTMB is a good example, because the detection limits are multidimensional, because it's a multivariate index score. What are your views on how to assess, or whether you need to assess LOD for BTMB?
I thought that is what we will be discussing in the pre-submission. This is like a really big topic to address, because of course there should be some amount of minimum metric needed, right, for any type of assay, whether it's going to be the same as how we have looked into the other assays, but there should be some amount of data point that we'll be looking into as what is the minimum metric that is needed to give a positive result 95% of the time. So yeah, there should be one needed, and how do you address it? I think that's what we have been discussing, and hopefully we'll get to an answer maybe the next time before we meet.
Yeah, I will say, I'm scrolling through the questions people have, and there's a lot of question about sensitivity. I think how to measure sensitivity, how to measure LOD kind of a top of mind topic for BLOODPAC participants, definitely. So one question we had was about NGS methods that report for SNVs percent of variant allele frequency, percent VAF, as their output. And because there could be some variability depending on assay to assay, has there been any push by the agency to report copy numbers of each variant per mil of plasma, or some other more kind of global assessment of sensitivity or number of molecules in a specimen?
Yeah, I think it depends on the assay. Yeah, there have been, I believe there are some assays were reporting copy numbers. And of course, for the NGS assays, most of them are reporting VAS. But I guess that's another metric that we can harmonize, especially for the MRD. What is it, whether maybe both of these are needed. What is the right metric for the MRD probably needs to be worked out. But I do see, different sponsors doing, sometimes they are doing one versus the other.
That's great. So fundamentally, it's going back to the assay developer to kind of say how their sensitivity is assessed and how their output is demonstrated.
Right, but if you have to do, like Julia was saying earlier, from a meta-analysis, you need to pull the data. So it has to be harmonized. Otherwise, it's going to be hard to pull that data set if you have multiple metrics from multiple manufacturers.
Absolutely. Okay, so now we have a question about the benefits that have been observed from IO therapies using TMB in addition to MSI and PD-L1 testing. So the question, which I'm not sure you guys are the best to answer, is how can Medicare and payers align on a monitoring approach that supports precision medicine, like with these multifactorial diagnostic tests coming into play?
Yes, I mean, that would be a question for CMS, and we don't take cost into consideration for our drug or device approvals at FDA, although realize it's an important issue.
Okay, and then how do you feel the BLOODPAC validation, analytical validation guidelines differ or align with the CAP CLSI guidelines?
How it with CAP CLIA guidelines, Jen, is that the question?
Yep.
And maybe that's a good question for CAP and CLIA, whether they believe it's really because we believe the validation protocol that we worked on is really well thought. It's from multiple stakeholders, and if someone follows those protocols, it should be a good analytically validated test. That I think, at least I have confidence in that with that protocol. So maybe that's something we could actually check with CAP and CLIA, whether this is very different from how it's with CAP and CLIA.
Absolutely. All right, well, I think we might have reached the end of our time. I'd like to thank you both for your time. Thank the BLOODPAC and the participants for supplying the questions. And I'll turn it back over to Elaine. Thank you very much.
Thanks, Jen. And yes, thank you to FDA and to Jen for your participation in this session. And now I'll turn it over to Lauren.
Thank you, Jen, Elaine, Dr. Phillip, and Dr. Beaver for providing us with such a thoughtful conversation. We are incredibly grateful for your time. Now I'm proud to share this next in-depth look at one of our key accomplishments, the BLOODPAC Data Commons. Developing a single library or warehouse for data generated by different liquid biopsy technologies remains one of the central motivating factors in the creation of the BLOODPAC Consortium. After 5 years of hard work, our Data Commons is now one of the largest repositories dedicated to liquid biopsy data in the world for clinical research and development.
The data that we generate per patient for a single blood tube is probably gigabases of data elements. Without proper insight derived from these data elements, it's pretty much wasted storage on the computer systems.
The BLOODPAC Data Commons is a resource for a research community so that they can share data to more rapidly advance research and, in our case, more rapidly have an impact on patients.
We laid out, with the help and guidance and advice of the FDA and other organizations like the College of American Pathologists, the bare minimum criteria that should go along with a tube of blood if the data set is going to be placed in the data commons.
BLOODPAC has data sets from its members. They range from trials to contrived data sets focused on liquid biopsies. In addition, we have a way for the broader liquid biopsy research community to provide data sets that we can host in the BLOODPAC Data Commons.
It helps accelerate the path from we have a technology that's working to let's have it validated and approved appropriately, and let's have it be accessible to patients.
Each year, as more and more stakeholders donate their data sets into a BLOODPAC Commons, I feel like the better and better we'll be at in giving some of that assurance potentially to a cancer patient on his or her journey.
That was a great look at the work and experts that yielded our Data Commons. Now, Dr. Philip Bebo, who will be presenting our final session about expanding accessibility. He is Chief Medical Officer and Senior Vice President at Illumina, BLOODPAC Executive Committee and founding member. But first and foremost, Dr. Bebo is a medical oncologist and physician scientist. Prior to entering industry, he worked in academia, including as professor of medicine and urology at the University of California at San Francisco. Phil, thank you for leading this important discussion.
Thank you, Lauren. And on behalf of the BLOODPAC's Accessibility Group, I do want to thank all of you for participating in our summit today. You know, it seems like a short time ago when we were in the Eisenhower building, previously referred to the old Executive Office building, or OEOB, during the ignition of Vice President Biden's moonshot, which is the origin story of the BLOODPAC. Now, 5 years later, we've seen the reignition of President Biden's moonshot. And as I reflect on today's program and the past 5 years, has seen remarkable progress on how liquid biopsies have become part of cancer care. Liquid biopsies now bring precision medicine to many individuals with advanced cancer, but without available tissue. We see two leading commercial providers of ctDNA tests with FDA single-site PMAs. There's a national coverage decision for NGS-based diagnostics that can include ctDNA-based assays. Many public payers have positive coverage policies, and we see the commercial availability of liquid biopsies for not only treatment decision, but both monitoring and screening of cancer. It's remarkable to me that as we consider the mission and activities of BLOODPAC 5 years ago, that so much progress has been made. It has been gratifying professionally to see the progress in BLOODPAC's impact. The progress is also personal. At Illumina, as a self-insured employer, we've decided to cover a multi-cancer early detection test to eligible employees. I qualified and was excited to get my cancer signal not detected result. I also had an employee share their story with me of a different result where a cancer signal was detected, a tissue of origin was provided, and resulted in the diagnosis of an early stage but aggressive cancer that he's currently under treatment with. So, and importantly, that cancer did not have another screening technology. So of course, these are two anecdotal stories, and we have a lot more work to do across organizations to understand the full performance and impact of these tests. We do see a very real initial evidence that there will be benefits. And as we've worked together, the BLOODPAC across academic, government, and industry, and a growing number of stakeholders, as we realize part of our mission and set standards for the development of high quality liquid biopsies, and as we've seen these standards adopted and support the commercialization of liquid biopsies, we understand that there are more challenges and increasingly are turned our attention to accessibility. So if we go to the first slide, with the promise of liquid biopsy starting to be realized, the executive committee of BLOODPAC decided that a major priority over the next 5 years must be ensuring that the benefits of liquid biopsies are realized by all cancer patients. We have committed the BLOODPAC to work across our membership and other organizations to identify and address key barriers to adoption with a specific attention on reducing disparities that are all too rampant in medical care. While our motivation to accelerate the availability of liquid biopsies predates the pandemic has certainly added more urgency to our purpose. The pandemic has resulted in 2 years of incredible suffering, loss, and disruption. We know there was a toll on cancer screening and cancer care. Colonoscopies, breast mammographies, and any elective procedure requiring a visit to healthcare facilities were dramatically reduced or delayed due to concerns over containment of the virus or the burdens of the healthcare system by those who are already sick by COVID-19. And as we know, cancer does not have a pandemic pause button. We did see liquid biopsies help address some of these challenges during the pandemic by bringing care to the patient, and physicians' interest in liquid biopsy has increased. The next slide demonstrates one of our BLOODPAC members performed a study, and they surveyed oncologists, 50 oncologists, to ask their perspectives on liquid biopsies. The study showed a consistent increase in anticipated use of liquid biopsies and a decreased use of repeat tissue biopsies during 2020, the first year of the pandemic and a year where we saw the biggest impact of the pandemic on healthcare utilization. While we all had barriers to access healthcare facilities during the pandemic, there are many patients who have difficulty accessing healthcare in non-pandemic times. The cost and expense of childcare, the cost and expense of travel, time off from work where they're not getting paid, create incredible barriers to many families. Thus, there's potential for liquid biopsies to address these barriers. The next slide shows how we're starting to consider the landscape of barriers. Liquid biopsies will need to address these barriers in order to ensure there's broad access. Barriers can include the introduction of any new technology into medical care. What is the data that supports that? Is there sufficient data supporting the clinical validity, clinical utility, and the performance of those technologies? Is the language used appropriate? There's a lot of social distancing in our communities. And are we providing these tests, the results, the education around it with the right language, with the right understanding of medical literacy? In some communities, there's incredible fears of discrimination. Or what is the impact of a test on an individual's family's finance? Not only the test itself, but what about the care that results from the test? And as technologies emerge, as the data we saw earlier today grows rapidly, where are healthcare providers and patients being educated about them? This is not an exhausted list, but our group will be refining this list through the specific lens of liquid biopsies for cancer care and committing to key activities to make a significant impact in addressing these barriers. And as these activities have urgency, because we know disparities in NGS testing already present. The next slide shows another study. I spoke earlier about the national coverage decision by CMS, and this coverage has absolutely increased the utilization of NGS-based testing for individuals with cancer. This study was published in JAMA Open Network, evaluated 93,000 patients with non-small cell lung cancer, metastatic colorectal cancer, metastatic breast cancer, or advanced melanoma. And the impact of the NCD had on utilization was quite clear. The study showed clearly having an NCT increased utilization across all groups. However, the increase in use was lower in Hispanic, Latino, and African American patients than Asian or non-Hispanic white patients. Additionally, individuals on Medicaid did not have as significant an increase as those on Medicare. And most importantly, whereas some of the disparities existed before the NCD, disparities were actually amplified by the NCD. And this is the key theme that we have to keep our eye on as part of the accessibility working group in the BLOODPAC. Liquid biopsies will improve cancer care and will improve cancer outcomes. However, we must ensure that liquid biopsies address and reduce disparities and not amplify them. So one of the questions we always ask ourselves is, how are we best positioned? Are we positioned to address this? Are we uniquely positioned to address this? With accessibility and our prior success, our growing membership, and our established groups do place us in a very good position to address accessibility. This table is a quick overview of the different established groups and how their activities can be directly addressed, address accessibility. We've prioritized key activities already that over a time period to address barriers in a way that we feel we're uniquely positioned. Through landscape, doing an exhaustive landscape of barriers to liquid biopsy adoption, standardizing the language, including the voice of advocacy groups, developing educational materials, targeting primary care physicians, continuing to advocate for positive policy coverage, and coming up with robust health economic models. Move the liquid biopsies further to the patient. Can we validate home testing? I saw a question on that already come through. And finally, ensure that BLOODPAC members who are commercializing tests come up with innovative ways to directly address the disparities. So in the final slide, of course, this cannot be done alone. And we have already great membership in BLOODPAC, great membership dedicated to accessibility in our accessibility working group, but we're still early in our journey, and there's absolutely opportunity for you to become involved. And to really wind down the day and really put a point on it, I want to introduce Rob Dumanaway, Director of Reimbursement Strategy at Thermo Fisher. Rob has over 20 years of experience in health insurance and diagnostics. He has worked for leaders in both fields, including his current role at Thermo Fisher and prior roles at companies such as United Healthcare. Rob fuses his decades of experience with the contagious ebullience that keeps all of us at the BLOODPAC energized as we work through difficult and challenging issues. He has also taken on a leadership role within the BLOODPAC on our reimbursement working group. Today, we've asked Rob to moderate a discussion between AJ Patel and Sandra Brown, a patient and a provider who have agreed to provide their critical perspectives on the importance of access and accessibility to liquid biopsies. Rob, take it away.
Thanks a lot, Phil. And thank you, Lauren, and to others associated with BLOODPAC for the chance to speak this afternoon. And let me just begin with a few comments. Starting with a real simple one. I think most of us are familiar with Ben Franklin, and I think a quote that fits well at this point in the afternoon, and that is, we must all hang together, or most assuredly, we shall all hang separately. I think the last couple hours have been fantastic in terms of hearing about progress over the last 5 years, the promise of the future, and partnerships around how we're going to get there. But I think, some of the things Phil just mentioned relative to accessibility are legit issues. And I am pleased to be joined by a couple guests this afternoon who both live this mission and, will certainly speak well from the perspective of both a provider and a patient. And I'll go through just some quick intros. Glad they've been able to join on screen, and we'll begin with the questions. First is Sandra Brown. Sandra is a genetic counselor and is the regional manager of genetic medicine at Providence St. Joe's in Orange County, Southern California. She received a bachelor's in molecular genetics from the University of Oregon in 2005, go Ducks, and completed an internship in cancer genetics at City of Hope. Also received a master's in genetic counseling from the University of California at Irvine. She's designed innovative programs to improve and expand the delivery of genetic services and believes in the roles of clinical genetic counselors in precision medicine fluency, population screening, genomic research, and personalized risk management and prevention. Welcome. And let me also introduce AJ Patel. AJ is a stage 4 lung cancer survivor, diagnosed in August of 2013, and was told he would probably not survive beyond 6 months. He's a legal analyst living in Southern California with his wife and 3 adult children. He is a fierce advocate in the lung cancer space and is a consumer reviewer for the Department of Defense, who manages the federal research grants for all cancers. So thank you both for joining, and very much appreciate your time this afternoon. Sandra, let me start with you, and AJ, you'll get the same question next. In addition to my intros, was hoping you could each maybe take a couple minutes, share a little bit about your background and experiences with personalized medicine. Sandra, if you go ahead.
Sure. Thank you so much for inviting me to speak and for the kind introduction. It's a big question, actually. I think one of the areas that we try to focus on the most is really equity, to make sure that when we're delivering risk assessment information, that we're delivering it in a way that allows for wide populations to have access. And we also provide a lot of education to make sure that our physicians, primary care physicians, all the way to our oncologists, medical oncologists and pathologists, feel like they're part of a team that is identifying patients who may benefit from precision medicine, from precision screening, precision risk reduction, really individualizing our care and outreach.
Thanks. And AJ.
Hi, Robert. My journey really began with a non-diagnosed symptom that I just did not know what was going on, but my regular doctors kept telling me, you're perfectly fine, you're healthy and keep going. It wasn't until I coughed up some blood in the summer of 2013 that I realized something was serious. A CT scan led to the diagnosis of lung cancer, which was unfathomable to me. But what's really remarkable is at that time, knowing nothing about medicine, I realized that this is a very serious condition, especially when I was told I have 6 months probably to live if I don't do anything. And the only thing they offered me was chemotherapy. There was no other course of treatment. And I thought there had to be something more than this that had to be going on in the 21st century today. So I decided to walk away from this facility and say, I'm not going to go through this. And they were absolutely shocked with me. And remarkably, I came across an organization and some individuals who pointed me in the direction of precision medicine that I'd never heard of before. And it's like having unfortunately, the analogy I'm giving is a war tool like a bomb, and it specifically targets its intended place to destroy. And that's what precision medicine is. And that was my first introduction to knowing about comprehensive genomic profiling. At that time, it was based upon a tumor profile, but now we've advanced incredibly so far. And the work of BLOODPAC here is, as I've been listening this morning, is making me so humbled.
Fantastic. So, and we'll stay with the same sequence, only some pun intended there, and we'll go with Sandra first. Could you maybe spend a minute or so talking about what your facility and what your practice looks like from both a patient and provider basis, a little bit with regard to, demographics and, kind of the work that's being done, and maybe just cite a couple instances of, some obvious barriers and, things you've done at St. Joe's to remove them.
Sure. So as the regional manager of genetics, I preside over 5 hospitals in Orange County and the high desert in Southern California, and also collaborate strongly with our Los Angeles hospitals, as well as with our Genomics Institute, which is Providence Health System wide, so that we can pilot and then scale the programs that we have that help to identify patients at risk, and then also to prevent any gaps in care. So one of the things that we do is to, for newly diagnosed cancer patients, we have a pathology screening program where every week we query the pathology reports to look for patients who are potentially high risk. We also have an immediate follow-up of those patients. We did notice that for germline testing, that patients who are sometimes diagnosed at very late stages, if they have ovarian or pancreatic, lung cancer is less commonly associated with germline risks. So, but lots of cancers can be diagnosed at these late stages. And we wanted to be able to get patients into receiving genomic information as soon as possible. So we developed a walk-in clinic where patients can just arrive and receive same-day services at our genetics departments. And that is something that we are scaling. The other thing that we did was develop a reflex testing protocol to automatically send tissue directly from pathology for tumor profiling. One of the things that we are now doing is creating a reflex protocol for what we see is about 25% of tissue that is insufficient for the tumor profiling that will go to instead of to like a solid tissue analysis, to go to liquid biopsy analysis. So our pathology screening program is also based in molecular screening. And so we screen molecular, and now we are looking at screening liquid biopsy results for evidence of germline. When we identify patients that are high risk, have a germline mutation, we also have what I call liquid screening. So when you talked about naming, I like to call it liquid screening because I think it's more accessible to patient understanding that if they're high risk with evidence of cancer, that what we're doing is liquid screening. So we have liquid screening research protocols. And then we also have population liquid screening protocols that are looking for identification of colon polyps as well as breast cancers.
Super. And, my guess is the next time this esteemed crew gets together, we could spend another half hour talking about just naming conventions, what's wrong, what's right, and where it should evolve. But we'll be back to you momentarily with regard to some of the barrier issues. AJ, I would say similar question to you, but based on your own experiences and, potentially associated with, some of the other patients you now interact with, what are some of the, memorable barriers you've seen to care and, specifically around testing?
For me, the actual barriers that I'm seeing, and I still continue to see, which is unfortunate currently, is just the oncology world not necessarily always understanding that there is more to the development of medicine than it has been in the last 30 years, especially the oncology space. And so not everybody is agreeing to understand that we need to perform comprehensive genomic profiling. And initially that would have been done with potentially, tumor profiling, but now we've advanced even further. And there's still this large level of disbelief. And I can understand in a liquid biopsy space that the disbelief is coming from, this is such precision targeting that can you really determine a person's type of cancer, the mutation, the drivers, and all the relevant medical stuff behind it. So I see that as a lot of skepticism when I speak to a lot of oncologists around the country, especially in the community space where they're so busy trying to deal with many, many patients, and they're not able to really absorb the new technologies and the new opportunities. And what BLOODPAC does when they standardize these things, it's going to add a lot of credibility and a lot of power, and most important, a lot, from my perspective, a lot of hope to patients, because going through a needle biopsy or an invasive biopsy is very, very stressful. It involves lots of hours, lots of travel, and lots of anxiety, and lots of risks that can involve depending on the type of biopsy they're performing, where rather they're performing it. So a liquid biopsy is really fantastic advancement. And I think patients and caregivers that I've spoken to are really ecstatic when they find out that there is this potential to determine if they have a driver or how their cancer can be treated from this simple test, which is the least invasive and least painful for the patient.
Got it. Thank you. And I'll, AJ, maybe back to you as far as barriers go. Oftentimes when we look at things like payer policies, it's kind of assumed that one test per one patient per lifetime is adequate. Assuming that's your experience is maybe just a little different, given the duration and successes you've seen personally.
Yes, yeah. The approach that one test in a lifetime is going to be fatal in many cases, especially in the cancer space and advanced stage cancers like mine. It's incurable, and you know, when I was originally told that I had 6 months to live, and you know, I thought there was no hope left, but here I am 9 years after being told this information, and I think, you know, payers are getting on board. My experience has been that they're pretty slow to get on board. I had a lot of challenges. I recently had a liquid biopsy performed myself. This was last year around this sort of time. And there was a lot of hurdles with the payers to make them understand this new technology. They're still, in my experience, haven't caught up with the medical advances that have been made. But there is a lot of assistance out there, and people who are in need of this type of diagnosis or test, there is a lot of assistance out there, is what I'm really grateful for. But it's not widespread, it's not widely known by many patients or perhaps advocacy groups. So there's a lot of work ahead of us there. And certainly with advanced stage cancers, things are in flux all the time. I mean, you know, you could be doing just fine one day, but you go for your next diagnostic testing and they've found new information which is problematic, and you may have to perform another test which may be a liquid test, and at that point in time perhaps there's even more advances in the type of profiling they can do. So it becomes imperative that the patient is given that access and is given that support by the payers, and that's a very critical piece, because the number 2 concern for a cancer patient is finance, beyond the diagnosis itself.
Sure, thank you. And, you know, Sandra, let me kind of throw it back to you. You know, back to this term of barriers. You know, your teams face extraordinary diversity and barriers on a regular basis, be it insurance status, ethnicity, languages spoken, socioeconomic status, let alone the issues specific to disease burden. How have you managed, and you know, what kinds of things do you see as primary blockers? And you know, how have your teams approached some of those challenges? Because I'm a huge fan of the consistency with which you're doing this across all patients.
Yeah, thank you. We have a chatbot that is looking at population-based well screening. And I was very concerned that the chatbot would not be well received by non-English speaking or primarily English speaking patients, or that there would be other barriers to participate with the chatbot. But we've found about a 94% completion rate from our patient populations. And I think a lot of that has to do with additional languages available in the chat, with the way that the chat goes out explaining what it is. And with our mammography centers, our well screening centers, the check-in, recognizing whether or not the patient completed the assessment, resending it to them, helping them to complete it. There's a lot that goes into that, but we do know that we're quite diverse in our patient populations. We have a lot of both Asian and Hispanic non-English speaking patient populations here. We are not primarily Caucasian Northern European. We're about equal to Hispanic in our with our Northern European populations, and we have high populations of Asian and Middle Eastern patients. And being in Southern California, there's a large immigrant population, so that we do have significant patients that are not primarily English speaking. So there are a lot of those kinds of barriers, but I think if you recognize them and have community outreach, there are ways to make sure that you're eliminating or easing, what we say is trying to ease the way of the patient. Having our clinics become same-day arrival walk-in clinics was the one thing that helped us, so that patients who may have economic issues with not having time off work, that they could just arrive right before maybe another appointment that they had nearby at their convenience. with our reflex testing program in pathology, we made a decision in our region to just test everybody and not worry about insurance coverage, so that we could provide institutional billing for patients that, or foundation support for patients that could not have insurance coverage. And then we utilize our industry partners who have patient financial assistance programs, and we have other patient financial assistance programs available as well.
Very cool. It's you know, really pleased with what you've shared there, and you know, kind of what that means both to the patient community, but dare I say to the physician and provider community as well. Yes. Because consistency of experience on both sides of the table is awfully important, and that's I was awfully excited to learn that. You know, AJ, you know, from your perspective, other areas that you're seeing today as it pertains to barriers, you know, access issues, you know, things that you absolutely want to see done differently.
I think, you know, one of the things that I'm proud to have been part of last year is lobbying the California State Assembly to make biomarker or comprehensive genomic profile testing as something that should be performed immediately after a diagnosis, and that it should be paid for by payers or insurance companies, organizations that provide the financial support to the consumers. And California adopted it, and I think it's a huge stride in the right direction. And I think there are a couple of other states around the country that have this bill in front of them, and I you know, urge everyone in the community and the population at large to support this. This is a serious disease for a long time now, and we made some huge strides in the last 30 years, especially in the last maybe 15 years with the advancement of precision medicine. And this is just yet another step forward with the liquid biopsy. And so it's very important that we take these barriers away. It's very important that we reach out to the communities, because there are underserved communities and people within those communities who know nothing about this, who are in a system such as a community hospital, and not everybody is aware of what's available to them. And if you don't know what's available to you, you don't know how to save your life. And my life was saved because I was able to navigate my way through something that I have no idea about, very complex. Oncology is, in my mind, one of the most complex fields of medicine that I've ever come across. I mean, I'm just absolutely amazed about all these people that we're meeting on this conference today, and my experiences in general of just how much effort is being going into this. But we need to make sure that we reach the consumer or the patient or the communities, because they're the ones who are really struggling in not getting the information and technology in their hands.
Much appreciated. And Sandra, before I kind of throw it back to you, because that's so those are big shoes to fill, would just ask, you know, Phil, looks to me like there may have been a couple of questions posed along the way. If you don't mind, you know, maybe reading and asking them to the team in a couple of minutes. You know, we'll kind of tag team, you know, the final few minutes of this segment, and you know, open things up should others have other questions as well. But you know, Sandra, maybe back to you. You know, some other thoughts or additional thoughts on what's broken, what needs to get fixed, where you see, you know, some of the best opportunities to deal with some of the access and barrier issues that exist.
And I have to say thank you to AJ. That bill in California really made us all celebrate here. All of our executives, physicians, everyone's just so excited that that's really recognized as a patient access issue that that bill has really addressed for our state. Another bill that's important is the access to genetic counselor bill that's in front of Congress today. And it's shocking to me, being an old genetic counselor, that we are still not recognized by CMS as providers I wonder what we do. And so having that bill before Congress right now, it really needs all of our support. Genetic counselors are right here on the front line, making sure that patients of all types, all abilities have access to truly excellent genetic services. And we spend a lot of our time providing provider support, provider fluency, education to our communities. So we do so much outreach, and we are so valuable of a provider that it's hard for me to know that we're still not recognized by CMS.
No, that's appreciate it. And Phil, as I turn it over to you, I do so with a more than a usual smile on my face, because this may be the first July 1st I can think of, because if memory serves, that's when this bill starts to take hold or begin in California. Traditionally, my sense is that's kind of the time of year that most within a health system walk around with frowns on their face, because you're having to train an entirely new load of residents and interns walking through. So it'll be nice to see others with smiles on their faces to welcome the beginning of July. But Phil, with that as a segue, what do we have in the way of questions?
Yeah, thanks, Rob. It's fabulous. Thanks, AJ. Thanks, Sandra, for your perspectives. Yeah, a lot of interest. There's a question from Christian Rolfo to Sandra. During your first comments about the testing, he asked, are you doing complementary approach at diagnostic using tissue and liquid? And then perhaps as a second question, what are you doing monitoring now?
I'm not a medical oncologist. So we are not, to my knowledge, we're not doing combined tissue and liquid, but our physicians are quite excellent in identifying when a liquid biopsy would be important for their patient care. And we are growing. The numbers of liquid biopsies that we're doing are increasing. We know that there's an issue with limited tissue or failed tissue that should not ever leave our pathology department for an attempt at solid tumor sequencing. So we are now creating a reflex where at pathology, those will go directly to liquid instead. And like I said, for our molecular pathology screening program, we're now working with our main industry providers in that liquid biopsy space to make sure that our pathology screening programs are still being maintained with the new kind of growing technology that we're using.
Excellent. And maybe, Rob, I think this next one, I'd love to get your perspective on. This comes from Jinming Zing. US payers have had very different coverage reimbursement strategies on liquid biopsy and companion diagnostics. What can be done to remove the coverage bottleneck, allowing physicians to order the appropriate test without worrying about coverage? It'd be great to get your perspective with your experience in the field.
Yeah, so it's a good question. I'll try and be somewhat succinct on this, but I think first off, there is tremendous variation even within the Medicare space with regard to how liquid biopsy is covered. I hate to say location matters because I sound like a realtor, but in 28 states, there's tremendous access to liquid biopsy to help with therapy selection in late stage oncology. However, in most of the rest of the country, that's not necessarily the case. Even in 10 states associated with national government services, who tomorrow introduces coverage for comprehensive genomic profiling, as enthused as we are about that coverage policy, it's really limited to tissue-based testing, unfortunately. So if you think that's diverse, the tapestry is even more involved as it pertains to commercial and other government payer scenarios. So first off, it's quite varied. Secondly, I think much of what we've talked about so far, the answer to that will generally lie in data. I think in a lot of different categories, certainly the bias to indications that have, frankly, more than 5 guideline-driven biomarkers lend themselves to NGS testing overall. I think most of the bias when liquid biopsy is covered has been towards lung because the time and clinical implications are drastic. The value of a biopsy and that information plays an enormous role with regard to, almost a dozen different, biomarkers and therapy selection issues. But ultimately, the short answer to that, if I'm capable of it, has to do with more data. We're pushing as BLOODPAC, we're pushing as a reimbursement work group, and absolutely invite others to join the fight and discussion with us around these areas.
Great. Thanks, Rob. I know we only have a few minutes left. I guess one final question for AJ, and Sandra, thank you for all your work. And I think AJ, Sandra's probably not your typical provider as far as her understanding of genomic tests. As you've engaged with providers yourself, as you've engaged with other patients who've had to navigate, what are your thoughts on provider education and where we are along those lines?
There's a lot of work to be done with provider education. I mean, Sandra is a unique person in terms of, in her space, she really does understand a lot about the issue with the oncology and the testing here. But there are far too many people or far too many organizations in this country, and regrettably, it's not any better, it's worse worldwide, because I've been assisting in the worldwide space with other providers around the world, and they just have a huge lack of understanding. There's a huge gap, and there is so much work to be done. So from a patient standpoint, I spend a lot of time talking to advocacy groups and spending my time presenting myself wherever I can to educate people, educate patients, because a patient that is informed becomes a dangerous patient to the oncology world or to their healthcare team, and eventually to the provider, which is where they need to really take on the advocacy for themselves to save their lives or to prolong their lives in a humane manner. And at the same time, we need to find more advocacy and education for the patient and healthcare, the caregivers, about the financial impact. So someone like Rob could be a great resource to help provide input on how patients can better their second most concern after a cancer diagnosis, which is finance.
Great. Well, those are the questions, Rob. Hand it back over to you to wrap it up.
Thank you all. Really appreciate your time for this afternoon session. Outstanding work. And with that, let's turn things back to Lauren and close this segment.
Thank you, Rob, Phil, AJ, and Sandra. We will continue to focus on accessibility with our final in-depth look at BLOODPAC's work today. Liquid biopsies have the potential to expand access. By educating patients and practitioners on the potential of the science, we can address and improve socioeconomic and geographic disparities to bring the best care to patients. How do we make technology and innovation accessible to everyone so that we're not just changing the lives of individuals who we run into today, but all of those tomorrow?
I think what we all recognize at the BLOODPAC is that liquid biopsy has the potential to address disparities, has the potential to bring best cancer care out of the hospitals, out of the clinic, and into the homes of individuals who have trouble accessing those resources.
Because you can get so much information out of a blood sample today, it does actually allow for a much wider population to have access to this type of cancer care.
There's actually a whole lot of data that's required for payers to say yes to things.
So there has to be a way that there becomes a body of data that can be acknowledged and agreed upon so that patients around the country have access to the technologies.
Clinicians and payers can look at a liquid biopsy test and say they use the BLOODPAC's guidelines for analytically validating this test. That's an additional piece of evidence that makes us comfortable that we should reimburse the test, that the test is ready for clinical use.
We're working with the FDA to understand the ins and outs of what it means to validate liquid biopsy.
Because it really relies on analyzing blood samples rather than tumor tissue, it's potentially a lot more accessible to patients in diverse care settings.
We're working through payer education. Where do we see gaps in language, in guidelines, and how can we work together to, directly engage payers?
How do we educate physicians? How do we educate the field on how they can best utilize what's been proven to be efficacious?
And then increasingly working with communities to understand the potential and the impact that these liquid biopsies will have, to make sure as these technologies get validated and have evidence to show that they bring benefit, that no community is left behind.
BLOODPAC is doing something together that we've not seen anybody do well individually.
I want to thank all of our presenters for an incredible afternoon of thoughtful discussion and for taking time out of their busy schedules to participate today. A special thank you to all BLOODPAC members, whether you were with us on day one or just joined us last week. We have helped BLOODPAC identify areas of common interest and collaboration among regulators, industry, patient advocates, and academia, bringing expertise, time, and collective experience to the table. We have been creative and willing to think above brand for the greater good, and as a result, our work will equip members of our community to solve the most complex challenges ahead. Most importantly, we are going to make people's lives better by honoring each data set and each tube of blood generously provided by cancer patients to accelerate approval and clinical use of these technologies. I would like to give a special thanks again to all of the members of the strategic planning groups. You've made today and the next 5 years possible. This has been an ambitious undertaking that needed the weight of the collective to succeed. Thank you all for joining us. I hope you will remain engaged or consider joining us in achieving our mission and audacious goals over the next 5 years. You can find us at bloodpac.org, follow us on Twitter at blood_pac, or keep in touch on LinkedIn. I hope everyone has a wonderful afternoon. Take care.